Hello, everyone. My name is Dale Sunny, and I am a part of the SI Bridges program here at Southern Illinois University, Carbondale. And I work in the physiology department, and my topic of research was, is the glucocorticoid receptor NR3C1 present in PIT1 progenitor cells? So for an overview, what is the pituitary gland? The pituitary gland is referred to as the master gland of the endocrine system because it controls the functions of many of the other endocrine glands. And it consists of two structures, the anterior and posterior lobe. The anterior lobe secretes the growth hormone, prolactin, thyroid-stimulating hormone, and many other hormones. And the posterior lobe secretes oxytocin and antidiuretic hormone. So for this experiment, we were looking at the proteins NR3C1 and PIT1. NR3C1 is a glucocorticoid receptor, and it's important for the growth hormone producing cells called somatotrophs. And PIT1 is a somatotrophic marker. It marks the cells which will develop into somatotrophs. And figure 1 here shows the different cell types in the pituitary gland. We have lactotrophs, somatotrophs, corticotropes, melanotrophs, and different other cell types. Figure 2 shows the development of the pituitary gland. This is basically showing the anatomy of the structure of the pituitary gland. And you can see how it's very similar to my results. The picture E14.5 in figure 2 is very similar to how my results are looking. And this is just basically for reference to show how they are similar to each other. And figure 3 is on glucocorticoid receptor signaling. So how it works is that the NR3C1, which is the glucocorticoid receptor, is already attached to HSP, which is called the heat shock protein, and it is in the cytoplasm. When glucocorticoid hormones come together, the NR3C1 binds to the glucocorticoid hormones and leaves the HSP, and it moves into the nucleus where it binds itself to the GRA, glucocorticoid response elements, and it activates gene expression. And the glucocorticoid receptor basically controls the genes that affect the growth, metabolism, and immune response, and it's expressed in almost all of the body cells. So one of the main reasons we are doing this is because a lot of people have growth hormone deficiency, and it costs a lot of money and health also to do these treatments. And a lot of them are just in trial basis. And one such therapy is called induced pluripotent stem cells, which is basically taking a differentiated cell from a person and de-differentiating it back to a stem cell, which will be changed into a cell that we want in the person and induced in them. So sometimes the body will not be able to differentiate between the cell which has been induced and the normal cell of the body. And so glucocorticoid receptor comes into play here, and we found out that NR3C1 and PIT1 could potentially work together and produce results. So in my lab, one of the graduate assistants, she did an experiment using dexamethasone, which is a glucocorticoid, and she did it on a mouse which was aged at 13.75 to 15.75 days old. And as you can see in figure 4, these were the results. You can see that initially they did not have any glucocorticoid showing, growth hormone showing. And in the next picture, you can see that there were several growth hormone positive cells present in the pituitary when it was exposed to the glucocorticoids. And this acts as a basis and foundation, which shows that glucocorticoids can definitely induce premature somatotropic maturation in mice. And going down to our results, my results were based on the mice which was aged at 14.5 days old. And we performed immunohistochemistry on them, and it's a process which takes 3 days to go. We add a lot of chemicals and colors to show if there has been any co-localization of NR3C1 and PIT1 together. And in the first picture of E14.5, picture number C, you can see that there has been 2 arrow marks, which indicates there have been co-localization of NR3C1 and PIT1. And you can see that we used not only E14.5, but also in adult mice tissue, which shows much more co-localization in mice, showing that NR3C1 and PIT1 were found in them. So to conclude this, we can say that we found NR3C1 that it's present in a subset of PIT1 positive cells at embryonic day E14.5 and the adult stages. And we can see that there has been more co-localization found in between NR3C1 and PIT1 in the adults than in the embryos, which was aged E14.5. For future research experiments, I would like to try this experiment on different ages to see to get other results also, probably at age E13.75 or E15.75, and hopefully come up with a different result which supports growth hormone therapy. And I would like to thank Dr. Lakshmi Bagdol, and also my mentor and graduate students who helped me throughout this project. Thank you so much.